Hello friends, and welcome to our last tutorial in Beginners Bioinformatics series. Let's learn some more fundamentals. In this tutorial, we will go through some basic information about phylogeny and phylogenetics, an exercise to explain the concept of phylogenetic tree, basic terminology used to describe a phylogenetic tree, and stages of a phylogenetic tree construction, tree building softwares and process. So let's begin. In phylogenetics, we study the evolutionary relatedness of phylogeny among living organisms. Molecular phylogeny refers to where we use molecular data to infer phylogeny. These phylogenies can be studied for either for various species or genes or proteins of interest by aligning them for comparisons. For example, comparisons of 16S and 18S r-RNA gene sequences from various organisms help the molecular phylogenetists to divide living organisms into three domains of eubacteria, archaea, and eukaryotes, rather than just prokaryotes and eukaryotes. A phylogeny can be expressed as a tree-like diagram. Nucleotide sequences evolve as mutations in DNA sequence keep accumulating, and rates of these mutations can be used in a mathematical approach to create an evolutionary model. Therefore, it is possible to create a phylogenetic tree by comparing the available nucleotide or protein sequences from various organisms, and these can be arranged based on our understanding of evolutionary model. First idea of showing evolutionary histories through a tree-like diagram was given by Charles Darwin in his book The Origin of Species. Let's learn phylogenetic concepts by an interesting example. Class species of vertebrates we understand includes fish, and fish are classified into three major groups: Agnatha with no jaws and no scales, Chondrichthyes which are jawed and cartilaginous fish, and Osteichthyes which are true bony fish. Now let's check some important morphological features here, like position of mouth is either ventral or on anterior tip. They have an exoskeleton or scales, or whether the tail is homocircle or heterocircle. Here you have a few different diagrams with their names. You have to study their morphological features as discussed earlier and assign marks to each of them based on the presence or absence of these features in this table. Once you are done, it should look like this. Now, based on the scores generated, we will place them next to each other, a process called nested clustering. Then connect the neighbors using lines which look like the branches of a tree. And here is the phylogenetic tree of 6 different fish based on their certain morphological features. Jenny, Joyce, and Joey belong to Osteichthyes or true bony fish group. Ela and Isha belong to Chondrichthyes group, and Linda was Agnatha with no jaws and no scales. This was an approximate demonstration how we can compare and score nucleotide and protein sequences from various organisms and create a phylogenetic tree based on their evolutionary relatedness. To understand phylogenetic relationship among organisms, we need to use molecular data. Since DNA is the universal genetic material except in some viruses and can be sequenced reliably, quickly, and inexpensively with the advent of next generation sequencing technologies, its use is most appropriate for molecular phylogeny. Moreover, nucleotide and protein sequences from different organisms upon comparison may show similarities and differences which can be converted into similarity scores. These similarity scores help in calculating branch lengths and their placement on the tree. Now let's learn some terminology. Phylogenetic tree are also called dendrograms. Dendrograms can be further cladograms or phylograms. Cladogram is a binary tree where any ancestral node can only have two branches, whereas phylograms are those cladograms which show different branch lengths. Phylograms are real trees and represent evolutionary distance. Topology, which represents the branching patterns of the tree. It has a biological significance as trees showing same topology will have the same biological interpretation. Branches indicate the flow of genetic information, and branch lengths indicate the extent of genetic change or divergence from the ancestral species. Therefore, longer the branch length, more divergence is indicated. We then align sequences in a multiple sequence alignment or MSA as shown here. Count the number of differences like human and chimp sequences here are 13 base pair long each and 2 bases are different. In this simple alignment, we could calculate the substitution per site for each set of organisms as shown. However, there may be multiple substitutions during the course of evolution that have occurred at any of these sites, which we have not taken into account in this simple calculation. To overcome this and other issues, it's important to use evolutionary model like Kimura's K80 or Tamura's T92 models to infer the overall genetic change that has occurred. What is overall genetic change? Is calculated as the product of the rate of substitution and the time that has elapsed as shown in the equation here. Nodes represent the points from where new branches emerge or old branches end. External nodes or tips represent the single terminal branches, sequences of which are used to construct phylogeny. The points where more than one branch meet are called internodes and represent the most recent common ancestor of all sequences in the phylogeny. A root represents a common ancestor from where all the branches originated. It is the oldest part of the tree and depicts the direction of the evolution. Phylogenetic trees can be either rooted or unrooted. If a common ancestor or an outgroup is known, a tree can be rooted at midpoint or as an outgroup root. Outgroup rooting involves more distantly related sequences to be included as an outgroup, whereas in midpoint rooting, we assume the presence of a molecular clock and place the root at the midpoint between the two longest branches. Otherwise, in an unrooted tree, branches are placed based on their relative similarities and differences. Now let's have a look at the process of tree building and which softwares are available for tree building. Phylogenetic tree building involves the following 4 steps. Step 1. Acquire a set of homologous nucleotide or protein sequences. Step 2. Perform a multiple sequence alignment to align these sequences. Step 3. Use of mathematical and tree building models to estimate a tree from the aligned sequences. And step 4. Bootstrapping or any other statistic to present the tree with high confidence level to clearly convey the relevant information. There are many softwares available for the tree building. Some popular ones are PHYLOGENY, PAUP, Mega, PAUP-UP, Dendropy, etc. In step 1, we acquire a set of homologous nucleotide or protein sequences by choosing a sequence of interactions from database like GenBank, SwissProt, etc. and run a blast search to find homologous sequences. Step 2. We perform a MSA to align those sequences. Multiple softwares are available for performing MSA, and most phylogenetic softwares also have an inbuilt MSA tool. Here the figure shows a MSA for multiple protein sequences which also have some gaps. In MSA, gap creation is adjusted and extensions are applied to optimize the alignment. Phylogenetic analysis is only performed to those regions of the MSA for which data is available for all taxa. Step 3. We use mathematical substitution data models to calculate base substitution rates. We take into account the rates of transitions and transversions, as transitions happen more frequently than transversions. For proteins, we use substitution matrices like PAM or BLOSSOM, which derive amino acid substitution rates from observations. The next step is the use of tree building methods, which could be character-based methods like maximum parsimony and maximum likelihood, or distance-based methods like Fish-Margoliouth or unweighted pair group with arithmetic mean, neighbor joining method, or minimum evolution methods. These methods use complex algebraic calculations to find a tree that best accounts for the observed sequence variations. The final step is to calculate confidence values for the tree. Since it's not possible to measure sensitivity and specificity of an inferred tree, as we do not know all the ancestral sequences and phylogenies, therefore bootstraps, likelihood, and Bayesian approaches are used to infer confidence. In bootstrap analysis, we statistically estimate the position of each branch by sampling 100 replicates. Bootstrap values are often shown on a phylogenetic tree. Here in this diagram, a value of 100 shows high confidence for the branch B, whereas a bootstrap value of 69 depicts less confidence for the branch D. Greater than 80% or 90% bootstrap support can be sometimes shown as an asterisk. So we learned some important concepts about phylogeny in this tutorial. If you have any questions and suggestions, please comment. Your comments are always welcome. If you have liked this video, please like and subscribe to my channel to keep me going. In this series, this was my last video. I'll be back soon with a new series on molecular biology basics. Do let me know if you want me to cover any other topics. Thank you and bye-bye.